Genome sequencing involves recognition of the nucleotides in a genome and determining their order of arrangement, and we see that this genome sequencing is actually evolved, and now the modern technologies of this genome sequencing has actually revolutionized the pace of scientific discoveries. DNA sequencing actually begins in 1970s with the development of Maxim Gilbert's method, and later on it got the pace by Sanger's method. So most of the modern day techniques are actually, they are kind of the derivatives from Sanger's method. Here we see the first published nucleotides which were published by Gilbert and Maxim. So actually they presented the first 24 base pairs of the DNA working on the LAC operators. So first genome sequence was actually RNA virus, that was MS2. It was sequenced by Walter Fier's group at University of Kent. So we can say that RNA sequencing was there before the DNA. I mean, the complete, as far as the complete genomes are concerned, the first of all sequenced genome was of an RNA virus. As far as the free living organisms are concerned, since we are in a discussion that so far viruses are whether living or non-living, so if we talk about the living organisms, we start talking from bacteria. So we can say the first among the living beings that was a bacteria was sequenced, that was Haemophilus influenzae. We talked about that earlier. So let's talk about MS2. MS2 is actually a virus that infects E. coli and Enterobacteriaceae. It's a single-stranded RNA sense. It has a sense strand. So actually in the host it gets duplicated, and where its antisense is formed, and from that the RNA copies are again formed. So it encodes an MS2 coat protein actually. So the entire nucleotide sequence was actually established by nuclease digestion and characterization of the fragment. So actually we have the enzymatic digestion of these nucleotides, and they were sequenced with the help of that. So actually in the total we have seen that it has 49 different codons, and that code for 129 amino acids, which actually it gives a long coat protein. So virus has just a coat, and then it has its genome, so it's just RNA and the proteins which are outer coat or covering. So here we see its genome. So we have the genes which are here, where for example there is a gene MAT that is actually helps in assembly, putting those proteins together, and then CP is actually a coat protein. We have replicase protein here, and then we also have a lysis protein that breaks the host cells. So we also see that CP, rep and lysis, this lysis gene is actually embedded between these two genes, so we can have the genes within the genes here. Here is the history of different milestones which were done while we are moving towards this DNA sequencing. So we start way back in 1870 with the discovery of DNA, and then we see in 1953 the major breakthrough is Watson and Crick model. We see here in 1977 we see this Sanger's method came, and worth mentioning here is Hood's name. So that actually gave us the automated sequencer, so that also changed the pace of technologies in this sequencing business. So Hood is actually working at California Institute of Technology. He invented the automated sequencers, which is actually the key technology. It was used in the Human Genome Project. We will talk about that in the next section. Here is Hood in a good mood, Professor Laurie E. Hood. So he worked at Institute of System Biology, Seattle, Washington. So in the end we conclude that genome sequencing involves the recognition and determining the order of the nucleotides in a genome, and advances in sequencing technology actually revolutionize the pace of scientific discovery.